Welcome back. We are discussing about photodynamic therapy, light-based therapy, in which drugs gets attached to specific cells, specific abnormal cells that we want to destroy, and these drugs upon activation by light produces reactive oxygen species mostly, and these oxygenated species oxidizes the cell to which they are attached to, thereby destroying the abnormal cell. That is the overall idea. Now, let us discuss little bit about the light that we are sending. Now, this needs to be accepted that firstly it is a linear process, there is nothing non-linear about it. So, it is a linear absorption, more light you send, the more light it gets absorbed, and it is a linear process going on, and we do not have mostly non-linear areas inside our body. There are exceptions, collagen is a primary exception, but overall it is a photosensitizer. The excitement of photosensitizer usually follow a linear absorption pattern, and it does not require a high peak power or coherent light source. Anything, if the drug is sensitive enough and if you have shown light onto it, especially in your skin treatment, when you are doing PDT in your skin, a lamp of correct wavelength can perform PDT. You do not require a very high peak power or a coherent light source. A lamp can work, or if you remember porphyrin or hematoporphyrin drug, they were special people who injected them, they were specifically asked to stay away from strong light or sunlight. They have to stay indoors in a dark condition for 4 to 6 weeks, because the drug was still available, still retained in their body. It takes a long time to get discharged, get excreted out, and they may cause damage. So, overall it is the sensitivity of the drug for a particular wavelength, and since it is more or less a linear absorption process, especially if the drug is applied externally to your skin, then you do not require laser light source. However, we use laser simply because it is monochromatic or it has a very selective wavelength, thereby it can be selectively targeted, and most importantly, lasers are targeted with optical fibers, and with the optical fiber what you are doing, you are not only delivering the light, you can make 2 or 3 optical fiber simultaneously together and put it near the area, and thereby extract some reflected light, some backscattered light, and analyze it to see what exactly is going on, meaning you can monitor the entire process of photodynamic therapy at real time, right. You know this, optical biopsy is happening, there are more than one fiber is sending a specific amount of light, and the light is, after hitting the tissue, is getting extracted. The extracted light is taken back by another fiber nearby, and that light is analyzed, and you get information about the properties of the area that light is hitting. So, why can't we do this here? Why can't we do this here? There are a bunch of optical fibers that are combined together in very thin tubes, few micrometer in diameter. The entire thing is less than few millimeter in diameter, and it is put inside some sort of through either a keyhole surgery or some through a orifice, and then one of them contain the specific laser light that excite the photosensitizer drug, and rest of them are simply monitoring, the rest of them are simply collecting the light and monitoring what is going on, monitoring the surrounding area, thereby you are overall monitoring the PDT process. Therefore, for all intent and purpose, instead of shining a torchlight in your mouth, we use lasers as optical fibers. So, this is a very real life example for this porphyrin. So, photophyrin is the copyrighted name of the drug the company sells, Porphyrin is obviously the compound, and which is the light sensitive photosensitizer, and these are the light dose that this is the real lab. So I actually got it from reliable sources, from NHS, I think, and this uses 630 nanometer excitation. Since this was the oldest one, all information regarding porphyrin is available over the internet. You can simply access it. Several of the other photosensitizer drugs are copyrighted. I am therefore putting the copyright thing so that there should not be any infringement afterwards. Several of these newer form of drugs, the entire details are restricted and not for public use. Mostly health professionals know them. Anyways, so these are the actual doses that you do. So if you are going for a surface treatment means outside the surface, and interstitial application means when it is inside somewhere, the cancer or the targeted cell is inside the tissue. So these are the exposure that you can do: 25 to 500 joule per centimeter square for surface treatment, whereas 100 to 400 joule per centimeter square for interstitial application. I think it is self evident, right, that why the energy per centimeter square is less when it is surface treatment, but more when it is interstitial application. If it is not clear, then I ask you to discuss this and come up with an answer. I will not be giving you all the answers, and this is pretty straightforward why you require more energy when you are sending light inside a human body. Maximum irradiance 200 milliwatt per centimeter square for surface treatment, 400 milliwatt per centimeter square for interstitial application. And the major problem that we are facing in PDT is the output power has to be 1 to 2 watt. Remember in previous one of the lectures when I encouraged medical practitioners or biotechnologists or even nanotechnologists to try to utilize this in your lab, some kind of a petri dish. The biggest deal breaker happened to be, apart from the drug of the cost of the drug, is 1 to 2 watt laser. Though this is not impossible to get, and several well funded laboratories do contain lasers which has 1 to 2 watt or even higher wattage, but 1 to 2 watt is not exactly the power that you get out of a normal laser pointer. So you have to have a slightly more effort, you have to show more effort to actually do the experiment. So this is the overall protocol. The protocol already exists for porphyrin, and different types of drugs are being generated which has similar dosimetry. Dosimetry is basically the dosage. Pharmaceutical students know it pretty well what dosimetry is, and they are asked to calculate. I know I have seen some of their syllabus, and dosimetry is probably one of their specific subject, specific paper. So this is the dosimetry for porphyrin drug for PDT. So all of this exists. All of this exists, and you obviously get dosimetry, or you obviously optimize dosimetry when it has been standardized or when it has gone through lots of trials and tribulations. So this actually exists. Rest of the time I am actually encouraging you to develop your own dosimetry by working on first petri dish and then move on from there to a mouse maybe, and then finally do preclinical studies and then go forward to treat actual human beings. So how do you deliver the light? Well, the compatibility of the light source with other clinical instruments such as endoscope and stereotactic devices, we need to, so thereby one of the main reason why we utilized optical fiber is because we wanted to connect an endoscope with it so that we could monitor the environment. How in real time, if you can monitor the tumor getting destroyed, the tumor getting burned, the tumor getting oxidized, we have an endoscope, an optical fiber connected with either relay lenses or these days camera to actually see, monitor the tumor in real time, and how much more drug you need to do, how much more intensity of light you need to produce. You can actually get a good look. We can actually monitor the entire experiment in real time, and we can see. So, the light delivery has to be something that is compatible with other diseases. That makes all of our life easy. It's, we do not you have to simply wait x amount of time and think that the tumor has simply reduced, the tumor has simply oxidized. We need to monitor it as well. Yes, this is the ability to continuously monitor light output and light dosimetry. This is part of endoscope, but in a more open sense, optical biopsy. You are sending bunch of light and collecting bunch of light, and thereby continuously not only just seeing, but understanding the chemistry change, right. Some of the light that gets scattered will carry the information of the molecules that it has scattered, the molecules that it has vibrated, and thereby we can understand basically the physics change as well as the chemistry change. Hopefully something else not have come up because of the burning. Hopefully healthy cells are not being burned, and we need to tailor the spatial distribution to match the tumor shape and size. So, this is your tumor and this is your light. How much of it is getting destroyed? Is it just this part, or it is the entire region? So, you have drug here and you have drug all over this place, but say your light is only inducing this biochemical reaction here. Will it be enough to destroy the entire thing, or it will partially destroy the tumor, or the light will not, since light is not able to go in the back corners, the back alleys, this part remain intact. So, those are basically the dosimetry that needs to be calculated, that needs to be optimized, and thereby what sort of light that you need to send, what wavelength, how much penetration depth it has on the specific tissue, all of that needs to be measured. Remember the course on the module on interaction of laser with biological material. I gave you a chart of which laser penetrates how far in which specific tissue. So, utilize that chart and try to see, and that chart will be helpful in determining basically what sort of wavelength of laser that you need to send at what intensity, what power density, etc., to destroy either different types of tumors or same tumor of different size. One is small, one is big, the same tumor, same type of tumor, small and big, or different types of tumors. So, these days we are mostly looking into non-linearity, 2 photon PDT. So, most photosensitizer works in 630 to 730 nanometer wavelength range. Remember, this is not exactly the window where the biological tissues are transparent. Biological tissues are more or less transparent. Well, by transparent I mean the scattering is comparatively less, from I think 630 to 1350 nanometer. So, you have an entire range where the tissue is scattering less. So, more amount of light could be penetrated. So, most of the photosensitizer work at this range, and here most of the laser penetration is 2 to 4 millimeter. You can increase the power of your laser, you can increase the intensity of your laser, thereby increasing the depth, but that might generate heat in the surrounding areas, thereby destroy healthy tissues. So, it basically defeats the purpose. So, the optimal energy of light, the optimal intensity that presently is working only allows it to go up to a depth of 2 to 4 millimeter, and that is pretty small. So, if you have a very internally born tumor, something inside head, and you need to excite it, your drug will go and attach to it, no problem. You have some sort of a antibody antigen complex, something you have formed, but how to excite it? If you are opening it up and then inserting the tube that far, you might go for a surgery anyways. you might go for a surgery anyways. So, that is presently the problem. Presently the bottleneck is that the tissue penetration is less than 2 to 4 nanometer. The PDT effect, well, we have seen 2 to 3 times deeper than that. 15 millimeter can be achieved, because as I said, the fiber does not necessarily need to go at the source of the tumor, as long as the light that is coming out of it is able to penetrate. You have used optimized light, you have used into a particular frequency at 15 millimeter, but still millimeters are small. Millimeters are small. We need something in the range of centimeter. If we are going for penetrative depth of tissues, tumors which are deeply buried, we need to go at a centimeter range, and that is these days developed by 2 photon PDT. That is developed these days by 2 photon PDT. How do you envision 2 photon PDT might happen? Remember a 2 photon microscopy, non-linearity, we discussed this thing in which 2 photons are coming and a third photon is coming out. 2 photons are coming into the input and a third photon is coming out into the output. Some sort of non-centrosymmetry happens. I am asking you as an exercise, I am leaving it as an exercise to look into non-linearity process, a 2 photon PDT. This is a very interesting and emerging field. I will give you all the hints that you need. You can ask me into the forum. So, overall it is a non-linear process. Overall you require a material which is non-centrosymmetric, i.e. which has second harmonic generation capacity, i.e. which takes 2 photon in the input and produces a third photon. The third photon has double the energy, obviously 2 omega. Omega plus omega gives rise to 2 omega. So, red lights can 2 red lights, 2 photons, 2 pulses can form blue light in the laser. We utilize it for tuning lasers, or we send infrared light, 2 pulses of infrared light can combine and generate frequency at visible range that has a slightly higher energy, and thereby it can penetrate, but you are not sending that high energy light throughout the entire process. So, this is the area where your tumor is, this is the area where the drug is. This drug is energized by 2 pulses. The 2 pulses converts a nonlinear process. The nonlinear process result in this drugs generating another photon. That photon is going little bit further to activate another photosensitizer, which is then connected with your tumor. It is a very new, and someone asked me what are the hot topics of research. This 2 photon PDT is a very hot topic in research. We have not yet been able to optimize it. There are numerous papers, I know, and if you are researching on this regard, kudos to you, but it is way too far away, way too far from standardizing or optimizing or actually showing very good results. And you can use high intensity IR pulse laser. So, less energy light can be sent, they will enact upon a photosensitizer material, upon a non-linear material. That non-linear material, upon reaction of these 2 sets of IR pulses, because of the non-linearity process, will produce a photon which is of far higher energy, and that far higher energy will go further and will activate another photosensitizer which is connected with a drug. So, it will allow around hopefully 1 to 2 centimeter as compared to 2 to 4 millimeter or even 15 millimeter penetration depth, and thereby we can target deeply buried hidden tumors. So, this basically is the overall light based photodynamic therapy. Light sources from I discussed light dosimetry and 2 photon PDT. I gave you enough hint on 2 photon PDT. I would ask you to study a little bit more on 2-photon PDT. It is quite an interesting topic, and we will continue our discussion further on various topics of light activated therapy in the subsequent classes. Thank you very much.